All living organisms are made up of cells. Cells were first discovered by Robert Hooke in 1665. Multicellular organisms can consist of tens of trillions of cells, and obviously, not all cells are identical. An organism develops from a single cell, and all cells basically inherit the same genome. The genome is the DNA containing chromosomes which encodes genes, many of which produce proteins when expressed. There are over 20,000 genes in the DNA of human cells, and we know that cell identities are linked to their gene expression profiles, that is, which genes are expressed and at what levels. Knowing this tells us a lot about a cell. Each cell has a set of genomic DNA. This DNA is like a large library of information, and each cell in our body has access to all the books/genes in this library, and depending on which books a cell reads and rereads, it will have a unique identity. Until very recent years, the primary method for studying individual cells was using microscopy, first applied by Anthony von Leeuwenhoek in the late 17th century. Since then, microscopy has advanced immensely, but nonetheless, using a microscope caps you off at seeing only a few out of tens of thousands of genes at a given time. Luckily, there exist methods to measure the expression of practically all genes at once. Sounds good, right? That would be a great help in characterizing all kinds of cells. The most popular method is RNA sequencing, but it too has limitations. With traditional RNA sequencing, you have to work with a bulk sample, which means scientists can only sequence or read the average gene expression across thousands of cells. Therefore, rare cell types will surely be masked by the expression of other cells. So to recap, right now we've got two established strategies to study cells and their gene expression, neither of which is perfect. And at last, there's a new frontier in biology which introduces single-cell RNA sequencing. Technological and experimental innovations, such as ways to isolate and manipulate cells, as well as the ability to work with extremely tiny amounts of DNA and RNA, have led to a breakthrough. We can now measure the expression of thousands of genes in an individual cell, and we can measure all that in up to millions of cells. This is truly revolutionary and was unimaginable even 10 years ago. These breakthroughs have sprung up many ambitious projects that aim to classify, tag, and thoroughly analyze every single cell in the human body. One such project is appropriately called the Human Cell Atlas. This project is a massive undertaking and would change science forever. Such projects would let us study rare and previously unknown cell types, peer into organism development with much more resolution, and characterize diseases in unparalleled detail. From child development to cancer progression, we will be able to see these things from a whole new perspective. As a very relevant example, the data collected so far by the Human Cell Atlas project is already being applied to the current coronavirus pandemic. This coronavirus targets multiple organs and enters cells having certain receptors and associated proteins on their surfaces. Scientists can now query the data to find out which specific cell types and which organs express genes encoding these proteins at high levels, thus gaining immediate clues as to the disease progression pathway. This would have been impossible even a year or two ago. Thank you.